So in this video, we're going to be discussing on vesicle movement from the Golgi apparatus to the other parts of the cell. So first, let's look back at COP1. Okay, so this is the coating protein that coats vesicles that move in a motion from the cis-Golgi network to the ER. So this is in anterograde motion. Okay, so the COP1's purpose. What is the COP1's purpose? So notice here, before we go to that as materials from the ER are carried in vesicles coated with COP2 into the cis-Golgi network, notice that how some of the resident proteins which bear a signal called KDEL are also carried in the vesicle. So it's not really a clean work. Okay, some of the resident proteins which are meant to be left behind are somehow carried within the vesicle that is coated with the COP2. So notice here, some of the purple proteins reach the Golgi network and they're supposed to be not here. Okay, so what happens is in the Golgi network, particularly the cis portion of the Golgi network, there are KDEL receptors. So these are the protein receptors that are able to detect the presence of resident proteins which are supposed to be returned back to the ER. So these KDEL receptors will bind to those proteins, okay? And the KDEL receptors with the bound proteins that are supposed to go back are going to group themselves together. And COP1 coats this portion and forms a vesicle that will be moving in an anterograde motion. So as you can see now here, we now have a vesicle that is devoid of the other proteins except for the resident proteins with a KDEL, okay? And it's gonna be returned back to the ER. Okay, so that's really the purpose of these COP1 coated vesicles. So they are also known as the recycling vesicles because they return back what's supposed to be in the ER, the resident proteins. So now let's talk specifically on proteins that are supposed to be headed to the lysosomes. Okay, if you can remember, the lysosomes are the digestive organelles of the cell. They contain the hydrolytic enzymes. Okay, and where are those hydrolytic enzymes made? Of course, they are made in the ER, and then they are transported to the Golgi network, and then from the Golgi network, they are supposed to be targeted to lysosomes. Okay, so there's the word targeting. So they're not supposed to be just carried anywhere. So these enzymes, hydrolytic enzymes that are supposed to go to the lysosome, have a signal attached to them called the mannose-6-phosphate. So in the form of an oligosaccharide again. Okay, so very important to have the signal. So this signal of mannose-6-phosphate attached to them will tell whatever receptor proteins that they are supposed to be brought to the lysosome. Okay, so that's the arrow here. They're supposed to go to the lysosome. So we can see here in this general illustration, we have here the lysosomal enzyme carried to the Golgi network. Okay, and then in the trans-Golgi network, we have a mannose-6-phosphate receptor, the yellow one here that looks like a tuning fork. It attaches to the mannose-6-phosphate in the lysosomal enzyme. And clathrin proteins will be the one to coat this because this is from the TGN to the outside towards the lysosome. Okay, so this is where clathrin works. And this vesicle now that's coated in clathrin and carries the lysosomal enzyme is going to merge with whatever it is that the cell has endocytosed. So like in phagocytosis. So if the cell has eaten something, okay, like a food, it's going to be in an endosome and it's going to merge with the lysosome or the enzyme here, okay, as we see here. And then the enzyme with the mannose-6-phosphate is going to be merged with whatever it is that it needs to digest, and now you have your lysosome. And the clathrin proteins and the mannose-6-phosphate receptors are going to be recycled. They are going to be carried back to the Golgi network, as we see here in step number 5. So now, enzymes for the lysosome are able to do their work because they are being brought to where they are supposed to be needed. And that's thanks to the receptors here, the mannose-6 receptors, and of course with the help of the clathrin coating for the vesicles. So here's just a detailed look of that. Okay, we can see here the mannose-6-phosphate receptor, and here's your lysosomal enzyme. And here you can see the clathrin coating. Alright, so now let's talk about the general targeting of vesicles to particular compartments. Okay, so first, the movement of vesicles towards a specific target compartment. How do vesicles move? How do they move in a path? Okay, because the cell cytoplasm is a very vast jelly. So how would a vesicle know which direction to go to? It turns out that cytoskeletons are very important. They serve as railways. Okay? So here we can see an illustration of a microtubule. And the vesicle is actually not just surrounded by the coating proteins, but there is also a motor protein that's attached to it, sort of like a leg. And with the help of ATP, the motor protein is going to walk the vesicle along this microtubule in a particular direction. So that's how vesicles move in a dictated way. They don't get lost in the cytoplasm because they follow the microtubule direction with the help of motor proteins attached to them. And of course, motor proteins work because of ATP. Okay, so once the vesicle is now near its destination, it needs to be tethered. Okay, so it needs to be tethered. So the tethering proteins here are sort of like the anchors. Okay, that a ship that's about to reach a particular port, there needs to be a tethering protein, an anchor, that will tether it on its target membrane. So notice here the tethering proteins. Okay, and we also have another set of tethering proteins here. Okay, and these are membrane specific, of course. And we see here the function of the GTP. So remember your GTP, the one produced in the Krebs cycle. Okay, it functions here as a source of energy. So once the tethering is done, the anchoring is done, okay, docking needs to happen. So the ship really needs to really dock on the port so that the passengers, the proteins that are carried in the vesicles rather, can move safely into the port, right? Okay. So the docking proteins are these snare proteins. So after the tethering, there's the docking. Okay, we have the V snares from the vesicle, so that's why it's called the V snare, and the T snare from the target membrane. Okay, they are going to stick together for docking. And so here we can see a computer-generated model of a vesicle that's surrounded by those docking proteins, the snares, and the tethering proteins. So in this case, this is a vesicle carrying a neurotransmitter in a neuron. Okay, and here we see an electron micrograph of the connection between two neurons. Okay, We can see here the gap. This is the synaptic cleft. And we can see here vesicles that are about to merge with the membrane here of this presynaptic cell. And you can see in these detailed illustrations here, or bigger pictures, how each of the vesicles here are connected with this sort of connection. So this could be the tethering protein. Okay, and here you can see more tethering proteins sort of connecting the vesicle with this target membrane. And of course, the snares are going to work after the tethering. And then the neurotransmitters can be released through out into the synaptic cleft and be detected by the next cell, okay, the postsynaptic domain. So here are the detailed steps. We can see here now a different view, a different style of drawing for the snares. Remember, these are the docking proteins, okay? So the snares sort of connect together, the V snare and the T snare, okay? And they sort of stretch or pull away, okay, at opposite directions, causing the membrane of the vesicle to sort of merge with the membrane of the target. And here, the membranes have fully merged. And now, the contents of your vesicle can go out into the target site. Okay, maybe in exocytosis. So, exocytosis is very important as nerve signals carried by action potentials, you've learned about action potentials, are carried into the synaptic terminal, as we see here. And then the vesicles are exocytosed, okay, the contents are exocytosed, and then detected by the postsynaptic cell so that the signal, the neuron signal, the electrical signal, can continue into the next neuron. Okay, so it's very important that the exocytosis here is triggered by a particular ion, the presence of calcium ions. And calcium ions are always important in exocytosis. One very important function of this will be in fertilization. Why is it that only one sperm cell can fertilize an egg, for example? Why can't the others follow? That's because when one sperm cell has made contact with the egg's cytoplasm, there will be a release of calcium ions that will cause these vesicles that are just resting underneath the membrane of the egg to be released. And the chemical that's released will be the cortical chemicals that will cause this membrane that we see here to form. So this is the fertilization membrane. This membrane is going to prevent other sperm cells from penetrating into the egg. So only the first sperm cell will be able to do so. Again, this is a result of an influx of calcium ions, exocytosis, as we see here as well. Now let's go back to lysosomes. So lysosomes are the digestive compartments of the cell. They contain hydrolytic enzymes carried to them via vesicles that are coated with clathrin, if you can remember. And these are some of the listings of the enzymes. So you can see here phosphatases, nucleases that are able to digest RNA or DNA, proteases that are able to digest proteins, and all of the other enzymes that are found in the lysosomes. Now, these enzymes only work in an acidic environment. So, the membrane of the lysosome actually contains a pump, okay, an ATPase that pumps hydrogen ions or protons up their concentration gradient meaning to say towards the inside of the lysosome. So there will be more hydrogen ions on the inside than on the outside. This is of course thanks to ATP's help. So this is active transport. So this is going to cause the inside of the lysosome to be acidic. And therefore, the enzymes here can work because it's an optimal environment for them to work. And here we can see a lysosome surrounding a mitochondrion that is about to be digested. So this mitochondrion could be damaged. It could be old mitochondrion that's about to be replaced by a new one. So one role of the lysosome is to do regulated destruction of organelles. So all organelles need to be disassembled, destroyed, so that new ones may be formed. And they can also be used to destroy potentially harmful foreign bodies, okay, like the ones that we saw a while ago in that detailed illustration of the lysosome merging with endocytosed foreign bodies. And when a lysosome digests some of the cell's own organelles, that is known as autophagy. Okay, so meaning to say the cell is willingly destroying in a planned way its own organelles to replace them with a new one. So here's the autophagic pathway. So enzymes going to the lysosomes, as we see here, carried with the clathrin-coated vesicles. They merge with a phagosome. Okay, so this is the phagosome containing the organelle that's about to be destroyed. And so they merge together and the organelle is destroyed. Okay, and some of the destroyed portions of the organelle are exocytosed, as we see here. Okay, and some of them remain inside the cell as pigmented chemicals we call lipofuscin, pigment granules. And this is a detailed view of how a lysosome works in the autophagic pathway. Now in the plant cells do not have lysosomes, but they have an equivalent, and it's the central vacuole. Okay? So the central vacuole is more than just an organelle used to store water or cell sap. It also acts sort of like a lysosome. Okay? So the central vacuole is surrounded here by a membrane known as a tonoplast. Okay? And the tonoplast actually also contains proton pumps that pumps protons into the vacuole, thus increasing the concentration of protons here inside. But not only protons, but also of other solutes. So in a way, the central vacuole will be very concentrated with solutes. And you know, with the movement of water always moves from less concentration of solutes to more concentration of solutes. So water will go inside the vacuole, and therefore the vacuole will increase in size, and therefore it will push the cell's other organelles to the side, and an increased sized vacuole will contribute to the turgidity of the whole plant cell. Okay, so it's also one way of plant cells growing, as we can see here. Okay, the vacuole increases in size, therefore increasing the size of the whole cell.